All right, so the webinar is now live and we can see some attendees filtering in. Putting it into gallery view for a second so we can see everybody's faces. Hey, hey. All right. Excellent. We've got 13 attendees, so I'm going to go ahead and get us started because we're at 3:01. Okay, thanks again to my panelists for joining me early and on time. Always a great feeling. So good afternoon, everybody. Thank you so much for joining us for part one of the Applied Ecology Minor Research Symposium. As many of you know, in addition to required coursework, every student who completes the minor must partner with a research mentor to collect and analyze data to answer a specific scientific question. This afternoon, we have 7 students presenting the results of their research. And just for context, I'm Erin McKenney, an assistant professor in applied ecology. I'm also the director of undergraduate programs, and I help organize the minor. So a really, really great, I think all the students who come through the minor are great, but I'm especially excited about the diverse array of students research that we're featuring this week. So before we get started, I would also like to acknowledge that North Carolina State University sits on land that was originally stewarded by the Tuscarora and Catawba tribes. We honor these tribes today by recognizing that this institution of higher education is built on land stolen from those who were here before the colonizers arrived. Additionally, this land has borne witness to over 400 years of the enslavement, torture, and systematic mistreatment of African people and their descendants. Indigenous Americans have shared their knowledge of the land and its resources since long before their first contact with European settlers, and have continued to play a vital role in the development of our local communities, North Carolina, and the nation. We honor these people today and every day by recognizing them in order to break the cycle of colonization and the continued erasure of Indigenous and Black peoples and other people of color. We must acknowledge the history of the spaces and places we occupy to both understand and unlearn the many ways that we have been socialized. So finally, before we get started, a few housekeeping logistics. Each presentation will be about 10 minutes long. We'll take questions after each presentation. So if you have questions, jot them down or just note them internally, or you can type them into the Q&A box, right? So after each presentation, if you want to ask your question out loud, you can raise your hand using the participant bar, and I will call on you and unmute you after the talk is concluded. Or you can type your questions into the Q&A box directly, and our presenters will answer them after their talk. If we don't have time to answer your question live, our presenters then have the ability to type answers to your questions using that same Q&A feature. Okay. All right. Let's go ahead and get started. Up first, we have Lydia Bjorklund, a senior double majoring in environmental sciences and marine science. Take it away, Lydia.
Okay. Thank you, Erin. I'm not muted. Okay. So like Dr. McKinney said, my name is Lydia Bjorklund, and I am a junior, senior in environmental science and marine science. And I'm excited to show you guys this research. I've been working on it since last semester, and I'm just really excited to share it with you. So I did research on juvenile blue crab long-term population trends. And first, I just wanted to start with a little bit about the actual blue crab. So they have a really complicated life cycle. The larvae start out in the open ocean, so they're floating around out in the open ocean, and they turn into little megalopa, and then the megalopa swim into the estuaries, and then when they swim into the estuaries, they'll settle into seagrass or they'll settle into shallow diatridal habitats. And then after they do that, then they'll metamorphosize into juveniles. And that's the stage that I'm going to be talking about today, is this stage right here. And in North Carolina, most of these megalopa will come in through the northern sites, like Oregon Inlet. They'll come in through here and then make their way down. So in 1999, there was a blue crab fishery collapse. And so what happened was there were 3 major hurricanes, Dennis, Floyd, and Irene. And those hurricanes caused extreme flooding, and that flooding caused a drop in salinity. So when all this fresh water came into the estuaries, it basically condensed the saltwater habitats. So there was a really small space for these blue crab juveniles to live and all the blue crabs to live. And that caused a massive migration to that small area. And fishermen were able to get a lot of crabs in that small little space. So there's a lot of overfishing happening. And you can see on this graph, you can see the year in the x-axis and the standing stock biomass in the y-axis. And you can see in 1999, there was a very large decline in blue crab standing stock biomass, and it's never fully recovered. So our question is, why has the population failed to rebound? And our hypothesis for that is the blue crab population has failed to rebound because of recruitment overfishing. And recruitment overfishing is just, you need a certain number of adults to replenish the population and make babies. But right now, we think the blue crab population doesn't have the right amount of adults to make the right amount of babies to replenish the population. So this slide is a little bit wordy, but just stay with me for 1 minute. We have a couple of objectives. So our first objective was just to measure the spatiotemporal distribution variability. And we have 2 hypotheses for that one. So the first one is juvenile density will change across sites. So we thought the Oregon Inlet and Mans Harbor will have greater densities than Hatteras Inlet and Inglehart. And our second hypothesis was juvenile density will change between the years. So from 1996 to 99, that will have a greater density than 2017 to 2018. And then our second objective was to compare blue crab juvenile densities in years before to 15 plus years following an overfishing event. And with that, our hypothesis was the historical data will show a higher juvenile blue crab density than the current data. So this part I wasn't here for. This was done before I came into the lab. On the left-hand side, you can see the map of the sites that the researchers sampled. So Mans Harbor and Oregon Inlet are more of the northern sites. And again, that's where those megalopa kind of come in and settle, and then they'll move down into this area. And they did that August, September, and October of each of the years they went out there. But the part I did in the lab was this. I would take those suction samples and I would sort through those and remove all the visible crabs and then get under a microscope, and that's this picture on the left-hand side, and slowly go through those and find the little baby crabs in there. And then I would take those, sort and count those, and photograph them, and that's this picture on the right-hand side. And then I would measure the carapace width using ImageJ software. And then with our data analysis, we did 2 things. We did, for objective 1, we did a 2-way ANOVA with interaction followed by the SNK test. And our response variable for that was juvenile blue crab density. And then our independent factors were year and site. And then with objective 2, we did a 2-way ANOVA with interaction. And the response variable was the same, the density. And then the independent factors were the pre and post overfishing event and site. So when we did that, this graph is for objective 1. You can see it's average of crab density by year. And on the x-axis, you can see all the years that they went out and sampled. And each site has a different colored bar, and each year has all 4 sites. And then on the y-axis, you can see average of crab density. And if you look at the graph, you can see that the yellow and gray bars, the yellow being Oregon Inlet and then the gray being Man's Harbor, they tend to be a little bit higher than the red and blue bars, which are Engelhard and Hatteras, all throughout the years that we did this research. So you can see that on that graph. And then on the left, you can see the ANOVA table, and those have the p-values for the year factor, site factor, and the year and site interaction. So when we're comparing the site and the year, we found that there are no temporal changes. The year factor showed no significance. The year and site interaction showed no significance. And if you remember, the p-value for the statistical significant p-value is 0.05. So if your p-value is under that, then that means it's statistically significant. So the only one that was statistically significant was the site factor. And once we determined that was significant, we did the SNK test, and that grouped the 4 sites into 2 different groups. And we found that the Oregon and Man's Harbor sites were consistently at a higher density throughout the years than the Hatteras and the Engelhard. And this graph is for objective 2. You can see the pre and post collapse comparison of average crab densities. And on the x-axis, this is the pre-collapse, and then we had the post-collapse, and then average of crab density. And this graph is a little interesting to see because you can see the pre-densities are a little less than the post-densities, and each bar is just a little bit less than the post ones. And then again, you can see on the left-hand side, the p-values, the site is the only one that was significant. But then this graph just shows it a little more clearly. You can see the pre and post collapse comparison by site, and you can see that difference very well in this graph. You can see the post collapse densities are a little bit more in each site than the pre-collapse densities, which, if you remember, is the exact opposite of our hypothesis. We believed that the post-collapse densities would be lower than the pre-collapse. And this was a really interesting time. We weren't expecting this at all. But with this, there were no temporal changes. The pre and post-collapse factor showed no significance. And then the site and the pre-post-collapse showed no significance. And the only factor again that showed significance was the Man's Harbor and Oregon Inlet. And you can see this very well in this graph as well. You can see these are both much higher than Engelhard and Hatteras. So there are a couple of take-home points. One being there was a higher juvenile density in northern sites, the Oregon Inlet and the Man's Harbor. And over the span of 20 years, Oregon and Man's Harbor sites had the highest juvenile density, which is really interesting because the historical data was from 1996 to 99, and then we did it again in 2017 and 2018. And you could still see that Oregon and Man's had the highest juvenile density despite the difference of time. Third, juvenile density post-collapse is higher than pre-collapse. And this was not expected at all. We were expecting the pre-collapse to be much higher. And then 4, if you remember, the standing stock biomass is still hasn't rebounded. So we're still continuing to see a reduction in crab abundance despite this post-collapse higher juvenile density. And right now, this is preliminary research, and it shows more juveniles now than it did before the collapse. So future research is needed to see if there's something happening between the sub-adult phase and the adult phase. And we were just speculating maybe it would have to do with overfishing or habitat destruction, but we don't know the answer to that right now. So more research is needed. And I would just like to thank Erin. This is my mentor. Her PhD dissertation is all about this. So if you have any questions about that, then you could ask her. and then Alyssa and Annika for helping me in the lab. And then this whole lab is Dr. Dave Eggleston's. So with that, are there any questions?
I don't see any questions in the chat. Again, panelists or attendees, if you have any questions, feel free to raise your hand and I can call on you and you can unmute and ask your question yourself, or feel free to use that Q&A button and type in questions as you have them. We have some comments coming in. Great job, Lydia. Yeah, got us started on a strong note. Awesome. I had a question. I'm wondering if maybe the collapse frees up resources that maybe fuels like greater fecundity in the remaining crabs. Do you think that's why you're seeing, you know, more juveniles?
Say that again.
So you were saying the number of the juvenile density post collapse was higher than you predicted. I wonder if that's, you know, if your crab population collapses and there are still resources available, maybe that means that the remaining crabs have more resources to fuel greater reproductive effort.
I don't know if that would be the case. We didn't look into that very much. I'll talk to Erin about that. That's interesting.
Yeah, and we've got a question from Alex Rocco in the Q&A. Why do you think that post-collapse densities are significantly higher considering the sorry state of the current NC stock?
That's a good question. So I don't know if it's just because of the complicated lifestyle that the blue crab has. So when they lay their eggs, they'll have the larva will go out into the open ocean, and that larva is out in the open ocean. So it's not in the estuaries where a lot of the overfishing is happening quite yet. So the juveniles will come back into the estuaries and they'll turn into, or the megalope will come into the estuaries and turn into the juveniles. So I don't know if it's just because they haven't been in the estuaries that they're not being overfished, and their densities are higher. I'm not sure.
I can see a couple other questions filtering in through the Q&A, but for now, if Lydia, if you could stop sharing your screen. And everyone, I think we should all have, oh, maybe we don't. You can, I was looking for my emoticons so that we could all cheer for you virtually. But we'll rest in confidence that everyone is cheering for you now. Thanks for getting us off to a strong start. Up next, and for our attendees, if you have questions, feel free to continue typing those into the Q&A. And we can see there are 3 more questions coming in, so feel free to type those in as you can. And up next, we have Yinan Ding, a senior in natural resources. So Yinan, if you can go ahead and share your screen.
Okay, can you see my screen now?
Yes, and if you click present, there you go.
Here. Okay. Hello everyone, my name is Yinan Ding. I'm a senior student at NC State majoring in natural resources. So for this semester, I've been working in J. Chow's lab with Matt Damiano on a new exploratory assessment of the common dolphinfish. What I did for this project is basically figuring out how to run the surplus production model in Excel and a relatively new method, JABBA, in our software. My main objective is to evaluate the effectiveness of using JABBA on this species. Okay, next slide. Okay, before I go on, I just want to briefly introduce some biology of the common dolphinfish. It's also known as mahi-mahi, and they are fast-growing species that can reach their sexual maturity within the first year. They are also highly migratory species that live in both pelagic and coastal waters between 20 to 30 Celsius degrees. So why do we want to study stock assessment for the common dolphinfish? Why this species in particular? First, due to their fast-growing and early maturity characteristic, plus they're tasty, and it contributes to the fisheries as a highly valuable and sustainable resource. So, however, it has not been well studied yet, and the allowable catch today is only based on the historical records. If you look at the graph on the right corner, this is a study done by Lynch and others in 2018. It indicates that the dolphinfish population is declining. However, therefore, there's just an urgent need for a formal stock assessment for this species. But a problem here is we only have very limited data about this species. So how are we gonna make this happen? We first tried to use the surplus production model. This model can be used to investigate the biomass dynamics of a stock, and it is particularly useful for data-limited species because it requires least parameters and data comparing to other methods. Using this method, we can answer questions about the current abundance, the fishing mortality rate, the historical stock, and the population changing influenced by the fisheries in the future. One thing to note here is that the surplus production model is an age-aggregated tool, which means we do not consider the age structure of the stock here. And this method consists of three main components: a population model, an observation model, and a statistical model. This is the first data we use to run the model is this aggregated landing data of each country in the Western Central Atlantic that reported to the Food and Agricultural Organization during 1950 to 2018. What we can see from this graph is that the overall commercial fishing, the gray line up here, is generally increasing over time. The second data we used is a nominal catch per unit effort data of each geographic unit recorded by the US pelagic longline fishery logbooks. This is also in the Western Central Atlantic. The data is ranging from 1991 to 2018, and the unit of the CPUE we use here is 100 hooks per set hour. This slide shows the results that we get from running surplus production model in Excel. So before we hit the run button of this model, we need to first enter in some guesstimated value for the population parameters, like here the KR and this Q, a catchability coefficient. And after applying some mathematical and statistical methods associated with those three models I mentioned before, it will give us the best estimate of these population parameters. However, one of the drawbacks of this model is that it only accounts for the observation error, which are the human errors that we can have in collecting data. So we need to use a more advanced tool that can give us a more precise model. That will lead us to the JABBA. JABBA is the acronym of the Just Another Bayesian Biomass Assessment. It is basically another surplus production model, but instead of using a frequentist statistic, we used a Bayesian statistic here. It allows us to incorporate what we already know into the model, which we call the priors or initial values. Here we have the K, R, and the new parameter psi. It is the biomass of the first year divided by K. The data required or we used here is the same one with what we did with the surplus production model, but JABBA can do more than just the biomass dynamics. It can also do retrospective analysis, sensitivity test, model diagnostics, and etc. Here with JABBA, we can also choose from 3 production functions: the Graham Schaefer, the Fox, and Pella-Tomlinson. One big difference here between the traditional production model and the JABBA is that with the surplus production model on the left, we assume the biomass maximum sustainable yield, MSY, or the maximum surplus production occur at the point where K divided by 2. The production curve is symmetrical around this point. However, in reality, this might not be true. So if we do not come up with a good MSY level, we are more likely to overfish to harm the recruitment potential of the stock. Therefore, in JABBA, we introduced a new shape parameter M here to better reflect the actual production dynamic. If you look on the graph on the right, with a smaller M value, the overall surplus production curve shifts more to the left, and the maximum is predicted to be reached earlier. These 2 graphs are the results from running the JABBA. We ran JABBA for approximately 15 times, trying with different data time ranges, initial values, and p-lim values. These 2 shows the result with different time ranges. If you can see, the catch data is from 1950 and the other one's from 1991. And these 2 graphs here are called the biplot. It can tell us basically how our fishery are doing throughout the period. It starts with this square shape. You cannot see, but it's here. And it ends with the triangle shape. These percentages on the top of each colored column represents the possibility of the fishery under each status. So the green basically tells us the fishery is doing fine and red is in danger. And the axis is a ratio of a fishing mortality divided by the MSY, and the y-axis is the biomass to the biomass MSY. If we look at this graph, from 1950, we can see that the fishery all of our fishing status is in the green column, but if you look at this gray area here, it represents the credibility. With on this graph, the credibility is pretty low. I would say it's 50 or even lower, but with 1991 data, the credibility is quite high. So we will just assume that the data range starting from 1950 has some problem with it. And this one shows the results with different R value. And this one is different K value. In a summary, with a higher R and higher K, there's more chances that we are in overfishing or overfished status. However, the credibility level is wider in both scenarios. Then we also played with a p-lim value. This value is the biomass divided by K, the carrying capacity. And our assumption here is that the recruitment potential becomes increasingly impaired below a given biomass ratio level, which is the PLM here. And if we pass the PLM, then we are preventing this population from recruiting. With all that, we summarize some uncertainties and limitations of the JABBA model. First, there are observation and process errors that we do not know, and we are not able to observe the spatial variance of the population because we only use a single data set that represents the overall dolphinfish stock. JABBA is relatively less sensitive than the traditional surplus production model, but still sensitivity is still a problem here, and it relies on people to use a very high quality data. Some take-home messages. Unfortunately, we cannot give any management implications out of this limited data situation. However, with the most credible data and priors, we used the projection graph on the right. Is the result from JABBA output. It tells us that the dolphinfish population is doing okay, and we don't have to be too worried about it. But again, this might not be 100% true for the reality. However, we do think that JABBA has the potential to be a good tool for the dolphinfish stock assessment, but needs some adjustment to the data and to the model itself. I also want to stress the importance of considering the environmental factors into the stock assessment, especially now the climate change is a big problem and the seawater temperature is rising. But here, we didn't incorporate any sea surface temperature data into our models. That's something we need to do in the future. And for any model, it cannot be exactly reflect the reality, but there are many good models for us to use in terms of having a better understanding of the population and maybe make a better management decision. So the most important thing for us to remember is that acknowledging just the limitation of each method so we can improve them in the future. Some future work we need for based on this project is that first we need to standardize our abundance indices, the CPUE data, with using a model-based method to reduce the process errors. And we also need to incorporate environmental factors like the sea surface temperature data. And last, we just want to make our model capable of investigating the regional variance of the dolphinfish population and their migratory patterns. The last, I just want to thanks to my mentor, Matt Damiano, and his mentor, Jay Chow, as well as those organizations who funded our research project. That's all. Any questions?
Thanks so much, Yinan. You have a question from Skylar Hopkins in the Q&A. Would you like to answer that live?
Do you have any recommendations for how undergrad students learn to use Bayesian approaches? So for me, I didn't know anything about Bayesian approaches, like Bayesian statistics, until I learned about JABBA. So I, sorry, I look at another question. So I don't have any recommendations. I learned from my mentor, but I believe there are many good resources online that you can go check it out. That's my answer. And how best way to cook a mahi-mahi? I do not know. I've only tried the mahi-mahi taco once, but I don't cook them myself, so.
Awesome. Well, thank you so much, Yinan. If you
Okay.
could stop sharing your screen,
Of course.
we'll transition to our next speaker. Up next, we have Jakob Fix. senior in animal science. And just make sure you're unmuted, Jacob, so we can hear you. Perfect.
All right. Hi, my name is Jakob Fix. As Dr. McKinney introduced me, I am a senior in animal science. And for my research project, we were looking at urbanization and environmental effects on raccoons in the Florida Keys. And I was partnered with Dr. Michael Coe from the North Carolina Museum of Natural Sciences in the Ecology department. And so just as a little bit of background, so raccoons are they're an omnivorous generalist species known for their extensive diet, of things which include mollusks, fish, crayfish, nuts, fruits, plants. And so they're known to inhabit lowland wetlands. But as the US has become increasingly more urban, we've started to notice that raccoons closer to developments start to take advantage of anthropogenic meals that are readily available, mostly from our trash and dump sites. This has led to raccoons being classified as synanthropes, which is an organism that benefits from humans in some capacity and are known to benefit from some artificial habitat. That being said, we do have areas still in the US where there's a lack of urban development. And as such, we see populations of these synanthropes without the urban development that they are supposed to be relying or gaining any benefit from. So in an area such as Big Pine Key in the Florida Keys, we see an area that has been protected due to endangered species such as the key deer. And here we can see a raccoon giving them a little peck on the mouth saying, "Thank you for keeping my area protected." But as such, we see that a species that's been present for hundreds of years has grown to develop an association with urban development, has an opportunity to experience the natural habitat. And we have a look at an area where that urban development was just beginning and then was halted due to conservation. So we have a rare glimpse at what how development can affect, or early development can affect species that we know for a fact will take advantage of anthropogenic meals. And so our research question became, how does development influence the occupancy of raccoons in Big Pine Key? And does our ability to detect these animals increase with proximity to urban development? Our big prediction was that we expected to see that even though there was much less urban development, we'll still see raccoons choosing to occupy spaces that are considered developed, right? And in this case, that means neighborhoods and community centers. This is mostly due to the availability of anthropogenic meals and them taking advantage of them. Our supplemental questions were, are there other environmental factors that play a big role in our detection probability, which we donated as P, and then our occupancy, which we donated as psi. And then we also looked at other synanthropes, or animals that rely on or have a relationship with human-based habitat and influence, to see if there's any correlation between those two species. So, so the study that the data that we analyzed came from a study that was conducted in 2013 that was looking into feral cat populations and their effect on marsh rabbits in Big Pine Key. That study utilized 24 square kilometers of refuge land in Big Pine Key, which was distributed among variable sized tracts and interspersed with urban private property and these community centers, which is what we denoted as urban development. The remaining area in Big Pine Key is largely protected under federal or state law. And so we established 112 sites with 2 pairs of cameras, and over 16 nights, we looked over presence and absence of different species in these areas. Fortunately for us, we captured a lot of different species, not just the feral cats and marsh rabbits. We had a lot of bycatch. And so we see animals like this opossum, and we saw lots of raccoons, which allowed us to analyze this data today and gain some inferences about what's going on in this unique area. So as a part of the initial study, they took measurements depending on the distance to the road and the distance to these developed areas, these private urban lands and community centers. They also took note of if they placed a camera on a trail that was donated at or marked as a human trail, meaning that humans frequently visited that location. For an environmental factor, we also looked at changes in elevation to see if there was any large difference in the areas that are occupied by raccoons or opossums. This was largely because we know that raccoons occupy these lowland wetlands, and we want to see if there's any sort of correlation there. And then briefly, I'll go over the presence absence that we collected. If you look at the trap night, we can see that there is on the first night right here, on the first site, there's a 0, which would mark that there's an absence of a raccoon at that specific night. And then if you look to the next night, there was a 1, which marks that a raccoon was present that night. And we can see throughout the data that if you look at, say, night number 5, you can see there's a distinct difference between the two sites where presence varied greatly, right? Which would then lead us to be able to compile this data. We ran it in a system that measures occupancy and detection probability called presence, which allows us to make inferences. So we look here at this table, we can see that we compared this data to several covariates, those including distance to the road, distance to development, whether or not they were on a human trail or elevation. And as we also ran this under the Ecology, sorry. The Ecology information criterion, which is a model that allows us to determine the quality of our model. The lower the AIC, the higher quality of the model. And so this table shows the top 5 models of raccoon occupancy, where psi again marks occupancy, and then P notes probability of detection. And so you can see our top model was detection with covariates of elevation and development. And then if you look at down the 5 rows, the bolded portion of the table recommends our null model, which is where no covariates affected either psi or P. And so our naive occupancy, our occupancy models showed a naive occupancy of 0.90, meaning that our desired species was photographed at 90% of the sites we used. And the data would suggest there's no variation in the occupancy over the research site. These models that we generated were linear, and what that means is that the probability of detection and occupancy, therefore the values that are reported are the beta 1 values, which is noted in that equation there, meaning those are the slopes of the lines, and those would note our correlation between the covariate and the detection of species. And so you'll look at the bottom left-hand corner, you'll see that there's a positive correlation of 0.11 that states that the further away we get from developed areas, we see a higher probability of detection of raccoons. And then in the top left, you see there's a negative correlation with raccoons in terms of changing elevation. And so we take these correlations, we notice that they're very small, but that being said, they're still, we counted them as significant as they fall within the margin, or the margin of error excludes 0, and therefore we determine them to be significant. We also looked at the opossum data, and we compared occupancy. We compared to elevation, and we saw that the slope of that line was 0.74, a positive 0.74, meaning that opossums were more likely to occupy spaces at higher elevations. And so the main thing that we started to infer was that with respect to distance and to development, we started to infer that raccoons likely were occupying, were avoiding urbanized locations due to the high density of prey in the lower in the less and then more wild habitat. And because there were less people back in 2013, there's probably a less amount of anthropogenic food available, therefore making the natural habitat a better choice for occupancy. And that's why we were able to detect them better. And our ability to detect raccoons and opossums depending on elevation could be indicators of one species influence over the other. It could be, we were seeing an instance of competition between two species over prey species, especially since they both overlap over a lot of different prey. Granted, we do look at this data retrospectively. It's 8 years old, so it may be worthwhile to reconduct the study and investigate the area and observe any changes in raccoon occupancy. For next steps, it would be helpful to assess raccoon density in this area, kind of gain a grasp of how many raccoons are actually present and what is driving them to occupy the lower lands. We start to also consider that the fact that in these areas, we face higher sea levels are beginning to rise. And so it's possible that raccoons might have moved from these lowlands up to higher elevations, which could cause interspecies competition between the raccoons and opossums. And we'll see that there's a variation between the elevations they occupy. Another thing that we'd like to look at would be for future research would be GPS tracking to establish a home range of raccoons and gain an understanding of how much time they spend in wild habitats versus developed areas, which could define the role that anthropogenic meals really play in the raccoon diet, or how much of a role we play in this smaller urban area. And finally, I'd like to give my thanks to Dr. Michael Cove and Annie Moore for providing me with the data that I was able to analyze, the North Carolina Museum of Natural Sciences for making this research possible, the NCSU Applied Ecology Program for giving me a space to share my research, and then the U.S. Fish and Wildlife Service for keeping Big Pine Key safe and protected. With that, any questions?
Awesome. Thank you so much, Jacob. Does anybody have one quick question that you want to ask live? Otherwise, I invite you to continue to, oh, did you name one? It's a popular question.
Affectionately, I named this particular photo in my computer Blair Witch because it reminded me of the popular movie. Just a very curious record. So if I had to say, there's one name there.
Appropriate. If anyone else has questions that you'd like to drop into the Q&A, please feel free to make use of that feature. And Jacob, keep an eye out for fan mail or questions about your work as we move on to our next presentation, okay? If you could stop sharing your screen, perfect. And thanks again for sharing your work. Up next, we'll hear from Hannah Cooper, a senior in marine sciences and zoology.
You see my screen okay?
Yes, perfect. You're good to go.
All right, hello everybody. My name is Hannah Cooper, and this semester I conducted research with Dr. Skylar Hopkins in the Sustainable Health Ecology Lab. And the work that I'll be talking to you about today centers around modeling the influence of animal movement on contacts. So in general, we use epidemiological models in ecology to understand and predict epidemics in human and wildlife populations. So you all have likely become really familiar with graphs that look somewhat like this shape. And this shows how preventative measures can flatten the curve of disease spread. So graphs like this are created using models that are built upon what we already know about diseases and what we know about the current disease and information from previous, and they're put together to create information and make decisions about management of epidemics. So when we're creating these models, even when we create human models, we oftentimes assume that animals move around randomly and make contact with each other randomly. But of course, we know that's not what actually happens in nature, and it's not how animals normally behave. So there are a lot of reasons why an animal might move non-randomly. but in my presentation, we'll focus on aggregation around food resources. So an example that you all are probably very familiar with is bird feeders. So when you put out a bird feeder, if you're very lucky, you'll have a lot of birds that will come and visit your feeder. One naturally occurring example of this phenomenon is animals gathering around a watering hole. And probably my favorite example of this occurring in nature is a whale fall at the bottom of the ocean. So in all of these examples, you can see that animals are aggregating around a food resource and are coming in contact with each other. So understanding how foraging behavior and how animals come into non-random contact with each other is essential to understanding how animals interact with their environment. Recent research also indicates that the way that animals are moving in relationship to their resources is changing with anthropogenic influence. So that makes this topic increasingly important to understand in the changing world that we live in right now. So in all the examples that we've explored, we can see that foraging puts organisms in contact with other organisms, and this creates an environment where diseases can spread. Additionally, stress from competition over resources may make organisms more susceptible to infection, so which makes it additionally important to understand how we can limit transmission of diseases. So a really valuable example to explore that demonstrates the relationship between foraging and infectious disease is in avian pink eye. So back in the 1990s, a bacterial pathogen made the jump from poultry to house finches, and the finches began to display signs of pink eye in the wild. A 2017 paper found that finches that spent longer times at bird feeders were both more likely to contract the pathogen and were also more likely to spread it to other finches. So this is a really excellent example and a really important example of how an organism's foraging behavior can directly impact the spread of a disease throughout the population. So the goal of my work this semester was to get a better understanding of the influence of animal characteristics and behaviors while foraging on the number of contacts it has with other animals. So overall, this will provide a better understanding of how diseases spread through populations. So the work I did this semester was based on experiments conducted in August 2019 on aquatic helosoma snails, which you can see along the bottom here. So a total of 56 trials were run where they varied the density of snails in each tank, and then also varied resources across 7 different distributions, which you can see in the picture on the left. So we'll explore more of the distributions on the next slide. So the snails were all monitored for 45 minutes, and during this time, their location on both resource and non-resource patches were noted, and also the size of the snails were recorded before the trials were run. So here you can see the 7 different distributions of resources that were used throughout the experiments, where you can see that the green patches represent patches where there are resources or where algae was grown, and then the brown patches represent where there is no resources. So it's really important that we vary these distribution of resources across the different experiments, and also so that we have a uniform amount of resource that is on the edge of the tank, because this can have an influence on how the organisms are behaving. So the numbers you see over top of each distribution is a number called the aggregation index. So this index is on a scale from 1 to 0, from most uniform to most aggregated in terms of edge habitat. So the work that I did was using information from experiments in the past, and previous researchers were able to use this information to develop a model, which you can see here. So you can see the snails are moving around. The black snails represent snails that have been infected. Healthy snails are represented in red, and then snails that have been infected and recovered are in blue. So you can see the movement here, and you can see that as the snails forage, the patches will turn purple. And you can also see on the left next to this patch area, they're counting the number of contacts and contact durations, as well as how many patches the snail has visited. So the work I did this semester was using this developed model and exploring relationships here. So an important question I wanted to answer was if the size of the snail had an impact on its movement. because if it did, this would be really important to incorporate into the model to get a better understanding of how this might influence the spread of disease. So I used a linear mixed model to analyze the relationship between snail size and the time spent on algae tiles from the data collected in that August 2019 study. So you can see here on the x-axis, we have the snail width ranging from the smallest snails to the largest snails, and then we have the time that they spent on an algae tile during the experiment. So this data was not significant, just as is. But then I wanted to dive a little bit further. And so you can see in this graph, we have the same setup where it's ranging from the smallest snail to the largest snail and also duration, but we have it split across the different number, like snail density across tanks. So we wanted to get a better understanding if the number of snails in the tank had an influence, again, with this the size of the snails in the tank, but this was also not significant. So even though neither of these aspects were significant, this is still really important information to have because we know now that we don't need to incorporate size into this model. So looking forward, there are still a lot of questions that I'm working on answering with using net logo modeling. One of the most important questions that I'm working on right now is if the size of the patch compared to the size of the snail has an influence on the number of contacts that are had during the trials. So each aspect that we evaluate in this model gets us closer to a model that can most accurately represent what we see in nature. This is really important in all epidemics and in human epidemics in making management decisions about how to limit the spread of infectious disease. So in terms of acknowledgements, I'd really like to thank Dr. Skylar Hopkins, who is my mentor throughout this project, Dr. Erin McKinney for her support throughout the extent of the applied ecology minor, and then Philip and Jeremy, who are two people who worked on this project previously and made the work that I did this semester possible. So thank you all, and I can take any questions.
Awesome. Thank you so much, Hannah. I can see you have at least one question in the Q&A if you want to answer that live.
Yeah, so the species of snails that we used, they were Helosoma snails. I can't remember the exact species, but Skylar would maybe have an answer to that. But one thing to note in terms of that is that while we used snails for this specific, like the trials that were run were with snails, in general, we're developing this model to hopefully be applied more generally to any population that could experience disease epidemic.
Any other questions for Hannah? You can also raise your hand if you would like to ask your question live. If a question percolates as we remain together, feel free to type it into the Q&A, and Hannah, you'll stick around for a little bit in case some questions come in. Well, thank you so much again. Now, up next, we have Lauren Willhite, a junior in environmental sciences.
Can we see this okay?
Yes.
Okay, awesome. So hi, my name is Lauren Willhite, and this research project was in collaboration with Crowd the Tap, and this research is called What's in Our Tap? An NC Pipe Investigation. So one of the main reasons why Crowd the Tap, the citizen science project that I'm working with, began was because of the many health hazards relating to lead and metal piping. And as we can see, with children, they are the most susceptible. They have adverse health effects dealing with development of the brain and nervous system, but this also impacts adults. It can have problems such as increased risk of high blood pressure and kidney damage, as well as these other things shown in the infographic here. Due to all of these health concerns that people began to get more aware of, the installation of lead pipes was banned in the US in 1986. But even with this, the replacement of the pipes in some water systems in some homes, much of the US like drinking infrastructure still has contains lead pipe service lines. So in the past, researchers have requested information from water providers on where these still exist, but the information that we have is only a partial picture of which areas and water systems still have lead service lines. So here in this photo, we can see the lead pipe disclosure policies and how well or not well they are. And so we can see where some of our holes in the data might come from. So the ultimate goal of the project is to create a model that will be able to identify who is at risk for lead pipe related issues and who isn't at risk, so that nonprofit organizations or water utilities or the local governments could use this data to hopefully lead to lead pipe replacement projects and prioritize who needs the most help. And then obviously it also helped me do my own research that we will talk about in a second. So how can this be seen like on a larger scale? An example of lead contamination on a larger scale is in the infamous example of Flint, Michigan. If you don't know the issues that surrounding this, essentially the city made a switch from supplying residents with Flint river water, and residents started complaining that the water in their taps looked, smelled, and tasted foul. Eventually, research came in and discovered that more than 40% of the water measured above 5 parts per billion of lead, which they consider to be a very serious issue. And in this photo here, you can see it caused some diseases as well, some disease outbreaks. And the contamination also exposed a lot of school children, and the effects are still being seen by the school system and parents. It causes like behavioral issues and just general development issues. But obviously this doesn't only affect people. It can also affect local ecosystems, including plants, animals, and soil, such as within like soil populations of microorganisms may be wiped out, which can slow the rate of decomposition, as well as populations of plants, invertebrates, and animals may be killed or hindered, allowing more lead tolerant populations of the same or different species to come and invade and take their place, which could lead to an increase in invasive species. So my research question is, what is the relationship between pipe material and home age within Apex? My hypothesis is that newer homes will have less metal piping compared to older homes because of the lead pipe amendment that I mentioned earlier. And therefore Apex would have less metal piping because it's like a newly developed area. So how is this research done? So as I mentioned, Crowd the Tap is a citizen science project. So a lot of the data is crowdsourced and will be used to create the model that I talked about earlier. And to do this, we have 3 general materials that we use. This first one is the form. This is one that participants fill out information about their pipes, homes, and they'll eventually submit their chemistry results up here. As you can see, some of the questions include what pipes is your foam for plumbing, what pipe material is the service line entering your home, and then there's these other tabs about just general information like the age of home, which I've focused on. This is the test strip. It's a slip of paper that you put into a cup of water for like 5 seconds, you leave out for 60. These colors will change and they'll correspond with this legend over here. This legend over here will tell you about the water chemistry, like it'll correspond to the water chemistry table, and it can tell you about alkalinity, pH, hardness, etc. Here is the bottle test. We do this because we like to get water that's at different parts of the pipe. So we have this first bottle is the first draw, so right when you turn on your water. The second one is like 30 to 45 seconds after you turn on the water. And then this one is 2 minutes after, so to get the different parts of the pipe. Additionally, we recommend that the participants wait to do this test until the water hasn't been used for 6 hours, generally right in the morning, so that the water can be still before they turn on the water. And so like if there's any leaching happening from it being still and sitting, then we can see that in the results. So here's a little bit of my timeline. On February 15th, I began my recruitment process. I started by posting to the Walden Creek Facebook page, which is the page for the neighborhood that I live in. I decided on this because I'm like a registered member of this and it's a private group. So I felt like they would be more likely to trust the project and trust me as I'm a recruiter. I want people to trust what I'm doing. And this is actually where I ended up getting most of my participants. Additionally, I also tried to contact the local environmental officers working at the town of Apex. I wanted to see if they were interested in the project, but emails were kind of slow. So nothing has come out of it yet, but I'm hoping potentially in the future that'll be able to work out. So from this, I've learned that recruiting from my own local community within friends and family also is the best strategy personally, because they ended up trusting the project more and were more likely to follow through with it. So we can see March 13th is when I had my first distribution of testing kits and bottles. And then I picked all those up and dropped them off to get sent to the lab on April 10th. I also picked up my second batch of kits so that I could deliver those. Now those have almost been finished, but sadly they haven't been delivered yet completely. And so we haven't had the results from those either. And even with this not being completely done yet, I've still been able to gather enough, resources and from the other ambassadors as well as previous data to complete the research. So here's my results. The first thing that I looked at was the type of pipe in all observed homes versus apex homes. So this light green here is the plastic pipes. Here is unknown. This dark blue is steel. This is copper. And then this cyan is lead. And then this right here is other. And then we can see between these two graphs that the plastic increases when you're looking at just the apex homes from 36 to 46%. And we can see that the this metal, the amount of metal piping, goes from this 8%, 25, and 1 to 0, 15, and 0. So we can see a little bit of a trend that apex might have less metal piping and more plastic piping. I did do a statistical test, the t-test to sample equal variance. The null hypothesis in this is that the numbers and the values are similar, or they have like kind of equal differences. But the alpha value was 0.05, and obviously this p-value is higher than that, so it wasn't a statistical difference, but I'll get to why it's still important later. So here, age of home in all observed homes versus the age of home in apex homes. This darker color here is 1986 or older or earlier, so this is like the older home category. This light blue here is the between 1987 and 2013, so like middle-aged, and then this is the newer ones after 2014, and this is unknown. Now between these two, we can see that the middle-aged homes for apex homes increases quite a bit, and then the older home category, it decreases from 35 to 4. So we can see that in apex, generally there are newer or middle-aged homes. Once again, the p-value was above the alpha value, so no statistical significant difference. Now for this slide, I looked at the home age within each type of pipe. So here we can see that older homes is 49% within steel, 57% within copper, 59% within lead, and then these are all the metal ones. But then within plastic, we can see that there's the 28% of older homes, so we can see the decrease within plastic. Whereas there's the big category of the between, like the middle-aged homes, and then we can see the newer homes are here as well. So on this page, we can kind of see that there's a general trend that within newer homes, or middle-aged homes, they tend to have higher like percentages of plastic pipes rather than the metal pipes. And once again, the values were above the alpha value, so not statistically significant enough. But what does this mean still? So as we saw in the graph, apex homes are newer and the types of pipes were mostly plastic. But within the metal pipes, they tended to have higher percentages of older homes, and plastic had newer homes. Now, even though the t-test failed to reject the null hypothesis for both of these, I think the results are still important because it can show us the general trend of what might be happening. In general, like that newer homes are tending to focus more on more plastic piping versus metal piping, which I hope is happening because that is a good trend to be going towards for safety reasons, as I mentioned before with health hazards. If we could get this to be on a larger scale research, then maybe we can find some more statistical significance, which we can get into with some future implications in research. What can we do to go off of these results? So the first thing that I thought was to continue this research in order to gain more participants and expand the sample size of the data. Currently, there's 995 results for all of them, but there's only 25 for Apex. So I think that if we increase the amount, we could look at it more like significantly and maybe use some different tests that would be able to more accurately be able to look at the data. The next thing that I would think is to do research within the apex area and compare the risk with an older area to see if there's a significant difference between risk in a newer and older area for piping or for just general water safety. And then the ultimate goal of the project is that once homes that are at risk are identified, hopefully there will be lead pipe replacement projects that can help with their problems. So these are my mentors, Deja Perkins and Dr. Cooper. I would also like to say thank you to Erin for helping me with this as well, and just in general. And then here are my sources, and thank you. This part where I ask for questions, if you have anything that maybe I can't answer, I put in the email for Crowd the Tap as well as the website about page.
Awesome. Thank you so much, Lauren. I was just typing your Crowd the Tap email address in.
Oh, if you didn't catch that, I can go ahead and put that in.
Oh, I typed quickly, so I just need to get rid of the extra C. So for anybody who did want to get in touch with the Crowd the Tap project, that email address is in the chat. And yeah, I'd love to open it up to questions. Again, if you are an attendee and would like to raise your hand to be unmuted and ask your question live, or feel free to type your question into the Q&A feature. It's for me always interesting to hear examples of when we actually want to move toward using plastic. I think, you know, in conservation and ecology, a lot of the conversation is about how do we stop plastic use, right? So now,
yeah, for sure.
Yeah, always a different mindset, right, with different goals in mind. Though certainly not single use.
Certainly not.
Well, thanks again, Lauren. I imagine that folks may be dropping some questions into the Q&A. So feel free to check that out and type in your answers as you have them. Skylar is sending you some fan mail.
Thank you, Skylar.
And up next, we'll hear from Alison Plumley, a senior in natural resources.
Sorry for the technical difficulties. The bar is covering up my.
Oh, you're fine. Your presentation is one tab to the right of the Zoom meeting. And that is an excellent workaround too.
Thanks. Wow.
I think you, yeah.
Sorry.
Nope. Totally fine. Good
Okay.
to go?
Yeah, can you guys see that very well?
That's perfect.
Okay, so just like she said, my name is Alison Plumley, and I am a senior in natural resources ecosystem assessment, and I'm excited to talk to you about my research on the influences of flooding on loblolly pine at Jordan Lake. So the study significance of this is that according to the Environmental Defense Fund, the number of extreme weather events has quadrupled since 1980. And for North Carolina, we are one of only 5 states in America that experiences this adverse weather. And that includes hurricanes, tornadoes, lightnings, fire, freezing, and hail. And although you normally think of hurricanes as being a coastal issue, as we learned from Hurricane Matthew, 92% of the damages that happened from that storm occurred inland. So our question, what can we do to mitigate this type of damage? And one of the answers that we have is that we can offset these extreme events by emphasizing our natural infrastructure. And this increases the land's ability to absorb water and to limit flooding and overall decrease the damage that these severe weather events can cause. So in North Carolina, one of the most significant infrastructures that we have is the loblolly pine forest. It provides food and habitat for more than 20 species of songbirds, for deers, rabbits, northern bobwhite, wild turkey, gray squirrels, fox squirrels, and that's just to name a few. Old growth forests have even been known to support the endangered red-cockaded woodpecker, even though that's not typically their habitat. But their loblolly trees are really well known for their ability to grow quickly in a variety of different environments, and they reproduce abundantly. So this makes them the perfect choice for restoration and environmental rehabilitation. So another special characteristic of loblolly, as you can see in this picture, is that it has the ability to grow in low-lying areas and in areas that are often flooded. But there is limited knowledge on how this affects their overall ability to thrive and grow in a forest ecosystem. So this leads us to our study question. Is there a significant difference in the growth and site index performance of naturally regenerated loblolly pine trees and those periodically inundated by floodwaters versus pine trees experiencing no flooding? So, and we hypothesize that the trees that are flooded will grow less than the trees that are not flooded. And as you can see to the right, this is our study area, and that is Lake Jordan. And in Lake Jordan is more than 4,000 acres of wooded area and over 14,000 acres of lake water there on that property that we sampled. So what we did was we picked 40 plots in these forested areas, and that's 5 trees per plot, and we picked them according to their crown class, which is dominant, codominant, and intermediate. And if you're unfamiliar with crown class, that just identifies the size of the overall tree, but especially the crown of the tree, the top. So pictured and labeled on the slide are some of the instruments and techniques that we use to measure the trees. And the picture 1 shows how we collect the tree height, which is just standard geometry. And in picture 2, you can see this instrument that's called a clinometer, and it just makes our job so much easier because it takes a lot of the guesswork out of it, and you don't have to do math while you're out in the field. So in number 3, this is somebody measuring the circumference of the tree. And we do this with what's called D tape or logger's tape. And it takes measurements in inches or centimeters and reduces them by the value of pi. So it'll actually give you the diameter of the tree. Pictures 4 and 5 show coring, which is done with an auger. And what we do is we take a cross section of the tree and we're able to extract that cross section with the auger and get a section that shows us the growth rings. And you may have, most of us are familiar with the growth rings, and that's how we count the age of the tree. So in the past, whenever you had to count these tree rings, it was kind of an arduous task. You had to sit there and count them. But now we have this wonderful software. I got you a picture of it, and you just put the samples on the scanner, you scan them, it uploads them onto a computer screen, and you can enlarge them, and it makes it really easy to either count them manually, or there is a program that will actually just count them for you and tell you the distance between each growth ring. And then it automatically stores that information in an Excel sheet, which makes it really easy to get the age of the tree. So whenever another data point that we collected was the height of the flood on some of these trees. And I thought this was really interesting because you see I've got the little arrows pointing to the pollen line on the trees, so you can actually tell where the floodwaters came up to on the trees. We learned that the most reliable way to collect this data is from the USGS flood areas, but you can see a lot of that pollen still left on the trees from flooding. So I have these, I created these whisker plots so you can kind of visually see what we're measuring. And when we measured the height and DBH, which DBH is called diameter at breast height, which is a standard 4.5 feet, where we measure the trees for its growth. And so as you can see on the whisker plots, we compare the crown class, which is, this is dominant, this is co-dominant, and then this is intermediate. So we don't compare them across, we just compare them to each other, which these are pretty evenly distributed. But we decided that to be more accurate in our predictions, what we really needed to do is to take into consideration the soil type that we were measuring the height and DBH in. So what we did was we separated them out, and this is just the DBH, because the DBH is really what measures the growth of the tree. And these are by soil class. And as you can see, once we started separating them, we had the GPS coordinates of each tree. And once we started sorting them out by the soil class, we found that we didn't have a full set of data. Some of the flood and some of the non-flood, they don't have matching data points. So they don't really give us an accurate representation of what the tree growth might be. So unfortunately, we had to reject our null hypothesis because we need additional data to truly understand the significant difference in the growth of the trees. And one of the ways that we plan on doing this is we're going to add some additional data points from the soil types and make sure that each soil type has a matching pair. And we have maybe add some additional plots to our study, even though 40 was definitely a lot of work out in the field, especially when it rained most of the semester, but we were still able to get it. And some of the area, another problem that we ran into was that some of the areas that we thought were non-flood zones, when we looked at the historical data, they ended up being actual flood zones. So we had to change some of our data, back from from no flood to flood. So, and I'd like to give my special thanks to Dr. Elizabeth Nichols. She was my mentor, and she helped me come up with this project, and Dr. Dennis Hazel, and he gave up a lot of information on how to do a site index, how to measure site index for the trees. And I'd also like to thank the TRIO Ronald E. McNair program for encouraging me to keep going through the semester and keep going forward with the research. And I'll be happy to take any questions that you guys have.
Thank you very much, Allison. Has anybody got questions? Again, feel free to raise your hand and I can unmute you if you want to ask your question live, or feel free to type your question into that Q&A feature. I'm playing Jeopardy music in my head to make sure everybody has a chance to process and type. Well, I'll invite you to feel free to keep filtering those questions in if you have them. And thank you again, Allison. And last but not least, we have Abby White, a senior in zoology. So Abby, if you'd like to share your screen.
Will do. I was just making a joke in my other class that I always go last.
Oh, alphabetical.
I think it's just my luck. Okay, do you have to accept me to, oh, found it, just kidding.
Nope.
I just the other day, I was looking for at the top.
You are already accepted.
So let me, yeah. Alrighty, can everyone see it?
Looks great.
Perfect. Alrighty, well, thanks for coming today. I'm doing my research on the effect of eutrophication on Pamlico Sound fisheries. Alrighty. So my introduction. So eutrophication is a growing effect or growing threat to the coastal ecosystem. So in case you're unfamiliar with it, it is the excessive loading of nutrients such as nitrogen and phosphorus in the water system. In other words, there is more algae present than what the ecosystem can support. So some direct effects are poor water quality, hypoxia, which is also just meaning low oxygen, physiological effects on fishes, harmful algal blooms, which you'll notice I say are abbreviated as HAB throughout. Indirect effects include habitat loss, decreased resource abundance, economic loss, and lack of domestic seafood. So the objective of the research were to assess the direct and indirect effects of eutrophication on commercial and recreational fishes in the Pamlico Sound. It's a little picture of a blue crab. So where specifically did we study it and why? So we did pick the Pamlico Sound because the Pamlico Sound does have little research of eutrophication. It is about 159 miles east of Raleigh. So as I stated, we were going to be researching the effects of specific fish species. So we had to find out exactly what species were located in the Pamlico Sound and then what species were actually affected by the effects of eutrophication. So as you can see, Pamlico Sound isn't far from Raleigh. All right, and then some of our hypothesis, using our knowledge on the effects of eutrophication in coastal habitats, we did predict that eutrophication does have the greatest negative impact on finfish species. So that we said, or we predicted that was because of fish kills, decreased optimal habitat conditions, and physiological effects. And then we did predict that oysters would be the least impacted by eutrophication because they do filter water. They can actually filter up to 50 gallons a day. And I'll get to more explaining on that one. And they are tolerant to hypoxia. So my research was probably a little bit different than most people's because I did a meta-analysis, which pretty much means that we and my partner reviewed a bunch of literature. So our literature that we reviewed consisted of fisheries economics, Pamlico Sound fisheries, economic effects on eutrophication, and the physiological responses of aquatic species. So pretty much what we did, I'll kind of sum it up in case you're confused, but we got our background information from like a broad variety of research on the fish and the area itself. Then we kind of narrowed it down once we found the species. So I'm going to go ahead to my next slide. Okay, so I just want to use this table to orient you the information that we compile. Of course, you don't have to read everything. This is just how we gathered all of our information. So once the information was found from a variety of sources, we made a table to make connections and gather the information. So we found species that were relevant to commercial and recreational fisheries in the Pamlico. So they do have a pretty big value within the Pamlico Sound. The gaps in what we could not find does show how much the species specific effects were understudied, and there is a lot more we could learn. So as you can see, like the Atlantic menhaden has a gap in the direct effects, and we also did mention like how well they were understood and the magnitude of the problem which in the Sound, which did allow us to pinpoint the specific species that were affected. Okay, so as far as the results, our paper, we do have like a long paper. This was the table that allowed, for all the information to be put in one section, so that way it was better understood. So this is pretty much like our sum of the results. So it does have the value, finfish being the most valued, and then the indirect, direct, and habitat that you'll find. So I'll give you a moment to look at that if you just want to go over it. Alrighty, so discussion. This is going to just sum up all the results. So blue crab, they did end up being the most susceptible to hypoxia. Because they are mobile, they are allowed to move habitats, which does decrease their habitat range also due to the SAVs, the submerged aquatic vegetation. Brown shrimp, they are also mobile, so they're allowed to move to shallow waters. Well, then moving to shallow waters does makes them vulnerable to predators and humans. Hypoxia also affects their growth, mortality, and catchability of fisheries, which I was talking about before is their physiological effects. The eastern oyster, as I said, they were tolerant of hypoxia and they can survive anoxia, which is what we found out. So that also means no oxygen. It does turn out that their main decrease is the over-harvesting and habitat destruction by humans. So let's see, in the shrimp, I forgot to mention. So about 51% of North Carolina's annual harvest of shrimp does come from the Pamlico Sound. So as you can see, eutrophication does affect the fisheries through there. And then the shrimp species are, of course, less affected because they're able to detect low dissolved oxygen and, like I said, avoid the areas because they are mobile. The oysters, so they do filter water constantly by removing pollutants and nutrients from the bottom column. And as I said earlier, they can filter up to 50 gallons a day. And then finfish. So finfish, fish kills, of course, is what they sound like. It's a localized die-off of fish populations. And as far as the fish kills go, in the US, menhaden comprise the majority of annual finfish mortality due to hypoxia. Benthic species, more tolerant but more exposed. So the benthic species are going to be the ones that are dwelling on the bottom. And then reduced where'd it go? Reduced reproductivity and lower spawning rates. Okay, so we did discuss in our paper, we kind of finished it by, putting the, what we could do to prevent hypoxia. So that's the mitigation of eutrophication. So the mitigation would be to reduce nitrogen and phosphorus, of course, which can be done by oyster reefs, reducing fertilizer and animal waste, and restoring ecosystems such as marshes and wetlands. So this picture is actually in Texas of an oyster reef. And then acknowledgements. I would like to thank Dr. Edwards for mentoring me through this research and my partner Alex for helping me as well, putting everything together. Excuse me. So this is actually a picture that my sister provided for me. It is located Pamlico, Morehead City. So it looks out at the sea, and it's recognizing the ones who lost their lives while working through the weather. And if there's any questions.
Thanks, Abby. Has anybody got questions? I'll again pull up that participants tab in case anybody wants to unmute to ask, or feel free to take advantage of that Q&A feature. Well, thank you all so much for joining us today. I hope you've enjoyed learning about our AEC Applied Ecology minor students research. Let's go ahead and give our presenters one last virtual round of applause. I know we can't all hear it. I need to like, yeah, you did it. Thank you for sharing all of your hard work. I think particularly through a pandemic, quite an achievement. And you can see your fan messages are coming in again. Remember, we do have 8 more students who are presenting on Friday from 3 to 5 p.m. So you can see the speaker lineup and the Zoom links at the Go link that I've provided in the chat. So we hope to see you back on Friday. Thank you again, everybody.
Thank you, Erin.
Yeah.